Hi, we are here right now looking at a poster of from Texas A&M University. This is Dr. Herman Sheltoff. He's a virologist working with antiviral silencing, and he and he's going to explain a little bit what his lab is doing and specifically what the poster is about. Herman, could you please tell us what's your research focus?
Yeah, thank you, Granty. My name is Herman Zulthoff, and as Granty mentioned, I'm interested in how do plants defend themselves against virus infection. And very recently, or not that very recently, but we've started to focus on RNA silencing. And in particular, how one of the components in RNA silencing is being used by plants against virus infection, and that's called RNA-induced silencing complex, or RISC. And we have published some information on that, how RISC might cleave the viral RNA. And a key component of the RNA-induced silencing complex, or RISC, is an argonaute protein. And we then asked the question, which was not known for plants like the solanaceous crop, not solanaceous host, Nicotiana benthamiana, that we're using here, which argonaute is being used. And a few years ago, we were able to obtain evidence that it was an argonaute that is named argonaute 2. It's a very arbitrary nomenclature, but it's an argonaute 2 that is being used against tomato bushy stone virus RNA. And we published that in Plant Physiology. And we then were interested in pursuing it further as to perhaps is argonaute 2 then a part of the RISC complex, to which we do not know the answer yet. But Veria, in her studies, has followed up on some of the argonaute 2 and other argonauts in her work, and that's what the poster is about.
And Veya is an assistant research scientist working with Herman. And Veya, please, could you tell us what today you have here in the poster?
Okay, so what we have here is, as Herman was saying, since we know that argonaute 2 is involved in the silencing of TBSB specifically, we wanted to study more. We wanted to study more what is the role of this protein in benthamiana during plant development, and also if it's induced by the virus when the infection process is occurring. So there is also another story that is related to this, that is has a relationship with the age of the plant and the ability of the virus to infect the plant. So we have noticed that when you use the wild-type form of TBSB, you get infection at different plant ages, no matter if there is any mechanism against this virus, the plant is infected. When you use an attenuated form of the virus that doesn't produce a suppressor of gene silencing, what we notice is that 2 weeks plants, the infection process occurs like if it's a wild-type virus. But then with age, you can see how inefficient is the virus in infecting the plant. When you look at 6 weeks old plants, we notice that there is no infection at all. Silencing is occurring very fast. So since we know that argonaute 2 is involved in this process, we want to see how argonaute 2 is expressed during development. So we went and looked at the argonaute 2 expression in healthy plants, and we compared it to argonaute 1 because we know argonaute 1 is a major component of silencing in Arabidopsis. But so what we see here is expression transcript levels by RT-PCR of AGO1. You see an increase in AGO1 with age of the plant. Here is for AGO2, and you also see an increase in expression, but the protein profile is different. We have Western blots, we have specific antibodies for these two, and you can see how AGO1 at the beginning of the plant development, it is not much expression, but this is only leaf tissue. We are not looking into meristems or anything. So you see how at 4 weeks, 5 weeks, 6 weeks, 7 weeks, AGO1 expression, protein levels are the same. But what we notice is that the AGO2 expression is actually decreasing with plant age. And this is a little bit contradictory with what we are seeing here in the plant development experiments with the infection, because if AGO2 is involved in this In this process, silencing, AGO2 in healthy plants is reduced at these areas. It's being expressed here, but then it's again reduced. And we see that silencing effects here. So now what we are trying to see is when you infect the plants with the virus, are you inducing the expression of Argonaute-2? So that's what we are doing here. In this panel here, you can see that the induction of different Argonauts under infection of TBSV, the virus wrap inoculated in the leaf area. And you see that there is induction of several Argonauts, but Argonaute-2 is a lot, many folds induced in the infection process. We also did the same to compare with other viruses, not only TBSV. And we noticed also that the AGO2, instead of the AGO1, has an induction of expression higher than the AGO1. So right now, we know the Argonaute-2 induces expression in presence of the viral infection. We know that when you knock out Argonaute-2, TBSV is able, the attenuated form is able to infect the plant. And right now we are isolating the complex, as Herman was saying, and trying to identify the other components and do Western blots to identify if Argonaute-2 is actually part of this risk complex or has an accessory activity.
Okay.
So,
yeah. Thank you. Great work. Yeah. Thanks.
you're welcome.